That's the last part of the final conference of GENE-SWitCH. And I'm giving this final talk to try to point as much as possible to give a message for achievements of the projects and perspectives for stakeholders. GENE-SWitCH has had, a quite ambitious global aim that was to deliver underpinning knowledge of the pig and chicken genome, and also to enable the translation of this knowledge to the pig and poultry sectors. And in doing this, in trying to achieve this, we had underlying questions about can we identify and characterize the role of functional genomic elements with an attention on those that are active, poised, or repressed during development in the determination of the phenotypes of the adult animal. And of course, what is the relative impact of the genetic variation of the functional and regulatory element of the genomes of the main production traits. We tackled these questions with a consortium, a quite small consortium compared to other European projects. You see all the members here. You have heard most of these people in the last 2 days. And in addition to the formal partners, we had also collaborations with breeding companies, in particular Aviagen, HyperBV, and the IFIP Association in France. And of course, since the beginning, GENE-SWitCH was part of the group of the first 3H2020 that was then joined by others as a FANG project, as really indicated by the Commission topic call. And the idea, of course, is the one that you know very well now. This is the picture that we show so many times on how as on the lessons of the ENCODE project in humans, assays by sequencing are used for annotating the different functionalities of the genome. And functional maps are useful to unravel which bits of a genome are functional, and this is, of course, pertaining different times, different cell types and tissues, different environmental constraints, in order to decipher the genotype to phenotype or G2P link. Sorry. I forgot to. In order to decipher the genotype to phenotype link. And this is, of course, fundamental for fundamental science to link cell, tissue, and whole animal scale knowledge. And finally, providing information to accelerate the genomic improvement. And on the bottom, you see the FANG global aims that are essentially the same. And the FANG initiative has worked in the past 10 years to standardize core assays and experimental protocols, to coordinate and facilitate data sharing via the FANG data portal, and establish suitable infrastructures for data analysis. And GENE-SWitCH, in this effort, has also contributed to co-build further developments. So here I placed GENE-SWitCH a bit in the history of FANG. So at the beginning, we had this paper published in 2015, the first white paper, where we were putting forward the concept about what functionally annotated maps are important as biological reference and for doing what, with a focus on main farm species, but not only, because FANG is always being addressed also in general to domesticated species, and starting with annotation of few adult individuals. In the so-called phase 2 of FANG, the idea was to extend this to different conditions like developmental stages, physiological conditions, environmental perturbations, and also additional species that at the beginning, 10 years ago, didn't have a so well annotated genome yet. And of course, trying really to invent predictive biology with new breeds, phenotyped populations, so not anymore just few animals, but also real populations to characterize. And of course, in this phase, it was very important to involve industry. And in 2021, we had this second white paper, the Fang to Fork concept, where some concepts were put forward on how to match the challenges of sustainable production for the next decade. And it is from this that actually there's been the development and now is ongoing project for a new research infrastructure in Europe that is Eurofang. And during the activities of GENE-SWitCH, we have since the beginning worked towards constructing these developments together with the other H2020 projects that started in different times. I mean, GENE-SWitCH, Bovreg, and Aquafang all started in 2018. Geronimo, Rumigen, Holoruminant started 2 years after, if I'm correct. And these projects could really make concrete clustering activities. I know that the Commission was quite surprised, but we were even exceeding expectations. And they had common features, like core activities of functional annotation of genomes of the different species, fish, bovine, chicken, pig. The aim to implement the phase 2, that is to use the functional annotations, like as it was in GENE-SWitCH, quite a lot of energies, to design new models for genomic selection. To co-develop with European industry and associations and organizations for breeding the research, so as really full partners, not just collaborations, and with consistent efforts in all the activities of dissemination, training, and networking activities. GENE-SWitCH, you heard quite a lot in the last 2 days. Most of you were here or could follow in streaming. In this picture, we have exemplified the concept and of the project. So we had the first aim that was functional annotations across tissues with a focus on different developmental stages in both chicken and pig. And you heard on November 6th p.m. the talks given by the colleagues mostly involved in these activities. So Alan, Archibald Herbert-Clock, Sylvain Foissac, Sarah Diabali, Martin Dirks, and Sébastien Guizard. In aim B, using functional annotation for precision breeding with the 2 different parts. We had the talks in the morning by Marco Bing, Mario Calus, Maria Ballester, Bruno Perez, Carl Johan Rubin, and Andreas Kranis for what is all around genomic selection and genomic improvement, and the talks by myself and Andrea Rau on the integration of epigenetics in this framework project. And all GENE-SWitCH outputs have been really conceived and carried out and obtained in the context of fair processes, findable, accessible, interoperable, and reusable. And all these outputs are really available to the community in terms of data, metadata, primary analysis, pipelines that are open shared in the GENE-SWitCH portal, in the Fang data portal, and in the dedicated page for GENE-SWitCH. And this data and metadata and so on are under the terms of the Fort Lauderdale Agreement and Toronto Statement, that is actually the data policy of a Fang action that has been accepted and shared by all these projects. We have scientific papers that have been collected under Zenodo. Otherwise, of course, in the normal research engines to find published papers and bioarchives and so on. Many, I would say, are still in progress. And if we think of these results and what could be exploitable for stakeholders, first, we had some exercise in discussing about this. And we think the estimated durability of the interest of these results is about 5–10 years, depending on what. And here I put again also how we have disseminated as much as possible all this by the website, other websites that are connected, including Ensemble and so on for the build, and the social media channels. So if we think of the first objectives, yes. If we think of the first objectives about the functional annotations across tissues and developmental stages, from the stakeholder perspective, we work quite a lot to collect a repository of tissue samples for pig and chicken that is stored at the Serbian IME of INRAE. And in here, we have samples. Some of them were completely used and are not available, of course, especially the samples from the early stages of development. Otherwise, we had made a biorepository of these animals. You see the list of tissues in the descriptions. And this could be available because it could be interesting for some groups in the community or whatever to say, okay, look, I would like to do some research on gonads or heart, and why not doing it on the same samples, on the same animals that have been characterized at all levels already by the project. In the process of assays, during the optimizations that was done and in the implementation, Diagenode, that is our biotech partner, actually also decided to develop a commercial kit for ATAC-seq. And this is commercialized for some time already. So in principle, customers can have it ready to use and easy, and it's one of the good kits, let's say, available on the market. And a very, very important outcome that actually contains also all the efforts in the scientific terms, the publications that we are making and so on. It's the first ensemble regulatory bills for these two species by MBL. So where all the genome annotation analyses are presented within an interactive genome browser. And what this means as benefit for the user is standardizations, reusability, lower bars for new research, pre-computed annotations, accelerate research and enable science, lowers economic and environmental cost of research. And the lead users and end users of this global achievement for this objective are academic and private laboratories, industry R&D components, and also the Eurofund industry, education, and finally also as end users, Eurofund research infrastructure. For the second objective, using functional annotation for precision animal breeding, this is quite a summary because, in fact, there was an incredible amount of work developed on this part. And you heard also yesterday the presentations of the FAIF, Functional and Evolutionary Trait Heritability Scores for variants in pigs. In this case, FAIF scores were evaluated for their potentials in pigs as a way to prioritize NIPS. And the benefit for users is that they provide a proof of concept to use these scores to prioritize NIPS in genomic prediction for pigs and also for other animals. And the lead users in this case are animal breeding companies, and particularly big farmers in this case. Another thing is that quite a big achievement of GENE-SWitCH have been new innovative genomic selection models that include the use of functional annotations that have been developed and also tested in commercial populations. And I think there is still a lot of use for these models in the now and in the near future. And This has led to several scientific outputs for evaluating the potential of using functional genomic knowledge to improve prediction, in which cases, for which traits more possible than for others. And I would say that this research has opened really a Pandora vase, because there is still a lot to discover on the use of these models and with new traits and so on. And the benefits, together with all the extension measures that we are put in place in Work Package 6, this has provided a proof of concept for the stakeholder community, and it also paved the way to the projects that followed, like Geronimo, that will make further use and, of course, improvement to this work. Lead users, animal breeding companies again, pig and poultry farmers. And finally, we had also this part of the project on the epigenetic of diets. And this has made most of all a proof of concept, also the difficulties that you can encounter that were really enormous, to implement a large-scale experimental setting to see effects that were so light and so faint, but still there, with some consequences that we don't know yet about what phenotype could, what at the level of phenotyping could mean in life of these animals. And these are data that can be exploited in further epigenetic studies, and we think it is most important for researchers and also for educational purposes. And then users again, researchers, industry, education, Eurofungery. Well, and then, where I am now? Voilà. About standardization of data and processes, dissemination and outreach. This is the third main objective. So here about what I already said, in fact, but it's fair resources. So protocols, metadata, data, analysis outputs. And you can see the description in the middle. So protocols that are available are made, are tested. They can be improved, of course, but this is the first standard, and as all standard in biology is evolving. But at least you have a process that is really controlled and can be followed by the community. Store metadata, raw sequencing, but also store primary analysis outputs for each experiment. Ready-to-use pipelines. A lot of work in GENE-SWitCH was done really on pipelines for bioinformatic analysis. And again, you see the potential benefits that are, I'm not going in the details again, in the benefits for users, and lead users and end users, more or less the same, in this case more academic and private laboratories, industry, R&D. And at the bottom you see also the other, a synthesis, Chagla, maybe not complete, of what is dissemination and outreach, so videos, glossaries, webinars, also with Eurofund research infrastructure, because at some point really these activities of dissemination and outreach could benefit of the collaborations and clustering that had taken place between all these projects. And these are support materials dedicated to a large audience, consumers, students, industry, professional actors, and so on. We think they are being already used, and they will be more useful in the future for training courses on genome annotations, genetics, genomics, and so on. And so it's a support for lead users like teachers, university lecturers, and users, of course, students, education, public. Overall, the main impacts of GENE-SWitCH are we promised them at the beginning of a project. It's high-quality reference annotation maps for the whole research community that I think has been achieved, in my opinion. Cutting-edge research results, paving the way to further studies and strategies. And even here, I think that we made our job. And ensure European stakeholder benefits with an increased understanding of the value of functional genome annotations to face current and future challenges to achieve sustainable productions. And I think also in this case, we have kind of made the job. Of course, many things could be done better, as it is always the case. I also remind that we had COVID after 6 months of start, so we had to face an incredible number of difficulties. We finished now in December after 6 months extension, that helped, but also complicated things for other matters. Anyway, I think we did all the best we could. And I underline that as additional impact, Eurofund and all these things that we built together, it's going to continue, provides a great opportunity for large scope genotype to phenotype research in Europe. And I would like to end here, and I give my final big thanks to all the participants in GENE-SWitCH for this adventure, and I thank you very much for your attention. Can I go?
So, hi, my name is Tatiana Zerjal, and I am with Frédéric Pitel, the co-coordinator of Geronimo. Geronimo stands for Genome and Epigenome Enabling Breeding in Monogastrics. It's a European project that works mainly on pigs and chickens. And there is a little transgenerational experiment in quails. This presentation will also be done by Marius, that will present you what is happening in Work Package 4 and the differences with GENE-SWitCH. So we are all aware that there are many challenges that we are facing: constant human population growth, environmental constraints. There are issues related to food security, and of course, a change in social cultural values. We are much more aware and conscious about the environmental footprints of our actions. So Geronimo, what hopes is to provide breeders with innovative genome and epigenome enabling selection methods to improve chicken and pig production, guaranteeing diversity. And this is done by the use of a large set of omics technologies, both genetics and non-genetic mechanism controlling traits, as well as the approach of a multi-trait analysis, including production, quality, and quantity, feed efficiency-related traits, productive longevity, fertility, resilience. Oh, sorry. I was changing. Is it like this? Okay, so it should work. It's true that it's hard to read from there, yeah, and it's hard to change, so. So a large set of traits that we called ELP traits to make it shorter. So it's a large consortium. We are 21 partners from 11 countries, and these include academic experts, private companies, professional associations, experts in innovation management, and expert in project management. And as you have heard several times, we are part of Eurofund. So the project is organized around 8 work packages. Work package 1, 2, and 3, which are in blue, are really, let's say, the data production work packages, where a large set of genetic and epigenetic data, mostly methylation data, will be produced. Work package 1, it's really trying to look at the genetic and epigenetic component affecting ELP traits. Work package 2 that Carlos yesterday mentioned briefly, it's really the work package interested in the environmental impact on epigenetic changes. Work package 3 looks at genetic and epigenetic diversity. Then we have two work packages, which are work package 4 and work package 5, where, let's say, analysis and models are proposed. So work package 4 will be Mario presenting it, and work package 5 will be Frank, and is the work package looking at social issues related to genomic innovation. Work package 6 is knowledge dissemination and multi-actor participation, and then we have the work package 7 for management and 8 for ethics. So the activity really, the idea is to combine the genetic part, so analyzing the sequence variation, and the epigenetic modification related to environmental changes. And in the project, we are really looking at transport and rearing condition changes and impact that can have at early life stages, feed quality, ambient temperature, age of the animals. So all these environmental changes and their impact on epigenetic modification to try to better understand the component contributing to phenotypic variation. So the project, of course, will lead to the generation of a large set of genetic and epigenetic data. And with this, the main aim of work package 1 and 2 is really to identify underlying biological mechanism affecting trait variation, and the work package 3 is to map genetic and epigenetic diversity on local breeds and propose strategies to optimize the conservation of genetic and epigenetic diversity. So what is new in Geronimo? Well, we think that there is a change of scale in number of animals and breeds studied. There will be, well, it's ongoing, massive genotype and methylation data production. It is a project that is focusing on a large range of traits related to production, asset, efficiency, but also welfare-related traits. And yeah, I repeat that. Well, it's looking at a large number of commercial and local breeds. What do we benefit from GENE-SWitCH? Well, the improved knowledge of the functional genomes will certainly enable us to go further in the understanding of biological mechanisms affecting trait variation. And when I say a change of scale in number of animals, well, it is an ambitious project because what we will produce is around 6,000 genotypes from chicken and pigs breeds. In terms of epigenotypes, so mostly methylation analysis, we are around 10,000, I would say samples, because sometimes we repeat several tissues. Transcriptomic, we are around 2,000 RNA-seq data, and the collaboration with several private breeding companies allows us to have access to more than 90,000 genotypes and phenotypes. And all this amount of data is needed for the analysis we will perform. So there will be a lot of GWAS going on with classic phenotypes, with gene expression phenotypes, with DNA methylation patterns in order to identify QTL, EQTL, and MEQTL for a large range of traits, as well as we would like to develop the EWAS approach. So use the methylation information plus classic phenotypes in order to identify QTM. So what is the impact of work package 1 and 3? I will let then Mario discuss work package 4. So we will implement high-throughput, non-destructive, low-cost phenotypes, implement a genome-wide, cost-effective tool to perform parallel genotyping and epigenotyping on a large number of individuals, identify biomarkers for early prediction of male reproductive quality in pigs and detrimental behavior in chickens, develop epigenetic toolboxes to identify whether animals have been exposed to detrimental conditions, stress, in the production environment, reinforce knowledge on genetic and epigenetic mechanisms underlying trait variability, and propose a set of options for conservation of genetic diversity and genetic diversity among and within breeds. As said, this large set of genetic and epigenetic data will benefit other work packages, like Work Package 4, to develop and validate methods to improve selection strategies, integrating functional information and non-genetic components. And I let.
Okay, thank you. So what I will do is I will explain in a bit more detail what we will do in Work Package 4 of Geronimo and how that relates or contrasts with what we have been doing in Work Package 4 of GENE-SWitCH. So in GENE-SWitCH, as also you've heard this morning and over the last few days, we've been generating assays, a lot of assays that cover the entire genome of pigs and poultry, and that says something at a population level of how important certain parts of the region of the genome are. And then we in Work Package 4 of GENE-SWitCH, we worked on using those functional annotations and genomic prediction. In Geronimo, we don't look at this population scale functional annotation, but we want to generate, or we are going to generate, individual methylation patterns. And then those methylation patterns and gene expression data we will use in genomic prediction models to try and improve that. So just for those that are not too familiar with that, to put it in a bit of a schematic overview, if we talk about genomic prediction, traditionally what has been implemented in the industry over the last 10, 15 years is that what you need is a reference population to do this. And this reference population needs to have a large number of individuals, let's say 1,000 or more. And for these animals, you should then have their genotype, so you should know their DNA, so to speak, or the DNA composition. And you should have phenotypes measured on those individuals for whatever traits of interest that are in your breeding goal. So here the example could be feed efficiency, and it could be, let's say, some growth-related trait or meat-related trait. Now, in GENE-SWitCH, what we then did is we looked at the genomes of individuals, and we tried to pinpoint, based on the functional annotation, where we thought is the relevant variation to explain phenotypic variation. So that's what these little red arrows on the DNA indicate here. In contrast, in Geronimo, what we add another layer of information for the reference population. So for these individuals, or at least a sizable subset of the animals in the reference population, we get an additional source of information, which we call other omics, because it could be, gene expression, it could be microbiome data, or it could be methylation data, like we are particularly interested in within Geronimo. So in terms of new models that have been developed within GENE-SWitCH, so there, what we try to do there is the traditional genomic prediction model just says, okay, we have 40,000 SNPs, we put them in the model, and then we just take the total genetic variance, we divide this by 40,000, and that's the amount of variance that each SNP can explain. And then we try to relax this assumption by saying, well, if we know that this region is functionally very relevant, we can upweigh the variance in that region. Or we're only going to select SNPs in regions where we know there's functional activity. In contrast, in Geronimo, in addition to using the genotypes of individuals in genomic prediction, we will use those other omics, such as the methylation profiles. And then the aim is to use that to help get a better grip on the variation in phenotypes and being better able to predict them, but also breeding values of animals. So that's just the take-home messages of this. Both projects aim to improve genomic prediction, and it's using newly generated omics or genomics data that's either at a population scale or at the individual level. And that's the connection, but also the contrast between both projects. And that's it. Questions? Wait for the microphone.
Sorry, I'm coming in the middle, so apologies. This is a very simple question about methylation patterns. How heritable are those?
That is actually something that we'll also be looking at within Geronimo, but I think from literature, there is much known about this. Can you say anything, Tatjana? Oh, you can take that.
So in Geronimo, there is one experiment with quails that will be a transgenerational experiment, and that will allow to answer the question. Right now, I don't have the answer.
And even if it would not be heritable, it could still be related to the phenotypes we're interested in, right? So it could, and it can still be relevant, yes.
I'm also a bit curious about how to interpret the results from GWAS on the methylation. So then you would find associations that impact methylation at a certain place. Is that?
Yeah, that's it. So try to find the genes or the regions that might affect methylation patterns, the GWAS, while the EWAS will be using the methylation data in order to associate it with potential. So it's the genetic of the methylation a little bit.
So they have a similar interpretation as EQTL in that sense, yes. Yeah.
I may have missed it, but all these methylation profiles, on which tissue do you have them? On blood or?
So because there are so many different phenotypes, we collect, well, the different experiments will look at different tissues. So what Carlo was mentioning yesterday, so concerning the behavior aspects, so welfare traits, he collected at least, around 10 different issues and many from the brain, in order to see if there are effects. We, on chickens, interested in so production longevity, because we repeated the sampling several times, we focused on red cells. In Wageningen, they are interested in fertility in males, and they work on sperm, so there are different tissues that will be analyzed. Any other question?
Hello, I am Donald Bruce, and I am on the Bulvreg project looking at public engagement, and I am not at all familiar with the field of animal genetics, so I'm an ethicist. But you mentioned, Elisabetta, the durability of your results being 5 to 10 years. Can you explain a little bit about what you meant by that? It was interesting to me just to how long the sort of effectiveness of this on production or on sort of development in general.
I don't know if I can explain much better than that. It's an exercise that we made in the XCOM with other people to try to really generate a synthesis of what we have achieved, okay? So we made these tables, and they say they must be maybe a bit better elaborated. But I showed them also because they were made, but it was a way also to not repeat again what was already said for 2 days. 5, 10 years, because, for example, 5 years is what? It's the full overlap there will be with projects like Geronimo and also others that we start after. And in scientific terms, we think it makes sense. On the other side, you can have other outputs that actually are longer because, for example, all these fair processes that we have implemented and all the standards that are already available for the community, in fact, they could even be more than 10 years. It depends on how you look at it, you see. So I cannot be more precise than that.
In the public engagement game that we're creating for Bulvreg, one of the actors is
Okay, use the microphone, please.
One of the actors in our game is a breeding company CEO, and he has the problem of, I am projecting 10 years ahead in what will happen in the cattle population. How do I choose now for things that will happen such a long time in advance? I just thought this is quite an interesting question as to how far these things will take to get into the production population, but who will be in and who will also be out.
But yeah, indeed, if you implement those things that we've found, and if people start to implement this in a breeding organization before it hits the farm, that will be 5 years at least, right? Because they have to make a change in the breeding program that will eventually change their genetics, but before their genetics actually arrives on the farm, that can take also several years, just because of the structure of things, yes.
So the question was complicated.
Ellen has a question. Ellen, please go ahead. You raised your hand.
So, yeah. So to attempt to answer Donald's question, if one considers genomic selection alone, it was conceived at the turn of the century, and it's continuing to have an impact on selective animal breeding to this day. And so elements of GENE-SWitCH were about refining that. So one could, I think it's not unreasonable to assume that those refinements will continue to have an impact in a decade's time. And in terms of the underlying reference genomes with their improved annotation, and those are underpinning research in the species where we've been working with. The human genome sequence is continuing to have an impact and inform genetics in that species, and I have no reason to doubt that the improved reference genomes that we've developed in this project will continue to underpin research in the species we've been working on for decades to come.
Thank you.
Thanks, Ellen. I think we have to proceed now to the next presentation by Frank. And thank you, Mario and Tatiana.
Otherwise, we get a nice presentation. Can I do your presentation? I don't know whether that will be so successful. But it would be a nice challenge of interdisciplinary cooperation. Yes, so after these nice presentations on what's going on in GENE-SWitCH and Geronimo, we actually make a bit of a switch in this session, which is also framed as a stakeholder session. And the idea is, of course, how to integrate different perspectives and also the more societal and ethical parts of what's going on. So what I would like to do with you is these 4 steps, actually as a preparation for the session we will do after the break. So the first is why is the attention to the societal ethical issues needed in the first place? To show that there is already discussion going on, but there are good reasons to broadening the scope of that discussion. There's also something that's going on in the 2 projects where I'm involved in Bovrek and in Geronimo. The importance of both stakeholder and public engagement, and more practical, how could we do that? So what are ideas of stakeholder and public engagement? There are a few examples in both the Geronimo and the Bovrek project, which is also something we will do after the break. Let's have a look. So you could say, why to start with the societal issues in the first place? So what you already noticed, I think, partly in the questions and answers here, but also in the presentations, already here we are together with at least a number of people who like so the back side, so the mechanical part, and if you are a watchmaker, then probably you like this. So the interesting things on cell lines, on QTLs, etc., etc. These have merits in its own sense. And these are essential to have discussions, good discussions about breeding and future possibilities. At the same time, for a lot of us, the backside of your watch is only to see how late it is and whether we are still in time. And I think that also holds for breeding. So the idea is that these innovations are most of the time an answer to all kind of scientific challenges, societal challenges. And just to give an example of the Geronimo project, you do not need to read it fully, but you already see. So when we designed the Geronimo project, we said a few things about what we like to achieve. And that's not only technical, not only scientific, it's about environmental constraints, about promoting food efficiency, about genetic diversity. And of course, we can have empirical discussion about, so what is genetic diversity, but it's suppose also the idea that apparently there is something important about genetic diversity, or there is something important about health and welfare. And that is where we make that step from the more technical or more empirical discussions to discussions that are very close to what plays in society and what we call ethics. The same as the claim that a number of projects, Geronimo included, says, well, we fit into the ideas of the farm to fork strategy. We try to contribute to the SDGs. Again, a lot of empirical discussion, but the whole idea, why should we be sustainable in the first place? That's an ethical question. I think maybe a lot of answers that will tell you why. But it's an important part to show that even if we have sometimes very technical discussions here, they are embedded in a societal context. That holds for I think a lot of parts in science, but especially in animal breeding, it's even more prominent. So I think breeding is by definition goal-oriented. I think the interesting discussion between Donald and Rita about the 5 to 10 years. You pursue certain things. You have certain aims. You have certain goals, and that immediately enters discussion, what kind of direction would you choose? What do you think to be important, and what is the preferred direction? Technology, in a very broad sense, but holds here as well. Technology is never neutral. You could develop a drone just to make pictures, and you could develop a drone as a weapon in a war. So that holds here as well. You could say, well, we're just looking at QTLs and these are very neutral. At the same time, you can make changes. You can make changes to the animal. You can do it for animal welfare. You could do it for production. And maybe it's not even that either or simple distinction. And that has implications in terms of who will be in power. What kind of uncertainties are you linked with, and what do you think is acceptable? And finally, and maybe that's a very specific part for the way of breeding we are looking here, it's about animals. And that makes, personally, I would say the discussion much more interesting, but at the same time, from a societal and ethical perspective, sometimes more complicated, because you see Europe-wide, but even beyond that, that the position of the animal is changing. So it's no longer just a thing that we can change to our will and how we like it. At the same time, we still do not have a kind of consensus within Europe to say, okay, well, this is the societal position of the animal. Still, it is something that you can own, that you can change. On the other hand, you see discussions about acknowledging animals' values or even rights. And it makes it complicated in this kind of discussion. So these answers are, as mentioned, never neutral. They're embedded in all kinds of discussion of what we think is desirable, is valuable. At the same time, and that may also a little bit reflect that we have a nice group here, but at the same time, we do not have a 150-person audience, because, yes, there is discussion, but it's not headline newspaper issues. So you could say, well, just leave it here. it's a nice discussion among us and don't make it too complicated. However, the absence of public debate doesn't imply that there are no ethical issues. On the other hand, I also would say, even if there is not a fierce debate, that should not be a reason not to include other stakeholders or the general public. You even could say it allows us to have good discussions and to include other stakeholders as much as possible to prevent that you end up in very polarized debates. Because we still have, for instance, in terms of genomic selection in BOVREG, in the literature already, examples of effects of animal welfare, mostly unexpected or at least not unintended. Questions about increased monopoly, so the idea is, are still farmers free to choose their own breeds, or are they becoming more and more depending on breeding companies? Linked to general needs for good practice for farms and breeding. So do you have codes within the professional field that helps us to enable the discussion? Then what we did also as part of the BOVREG project together with Koen Kramer is to say, well, apart from very specific ethical discussions, it's sometimes good to have a step back and say, well, start from a kind of three scoping questions. So the first is, which ethical concepts do you think are important to include in the discussion? So what you still see is that a lot of the discussions is framed in terms of risk and sometimes a bit of animal welfare. And risk is the idea is that if what we develop is safe, then it's okay. Well, the question is first of how to define safety, but even risks, how important they are, don't deny that, is a too limited frame to have discussions on what we do in terms of breeding. And that holds the same for animal welfare. If we discuss our relationship to animals, welfare is a very important part, but not all the issues that we have in terms of our relationship with animals can be reduced to animal welfare. A few examples from the literature. First is a review on genome editing in animals, where it said, well, animals can be harmed in other ways. The EU report on genome editing comes with the concept of de-animalization. A paper by Koen and myself, we talk about the concept of telos, and in the Geronimo project, we also look at concept of empathy. It's not that you have to capture this all directly now, but it's just to show that we have a lot of different concepts, and that's not ethicists who come up or design these concepts, but these are also quite often ways to capture intuitions we find in public discussion, so that people say, yes, it may not really be an effect on the animal, but we change the animal and we make it not into a being that we value, but much more as a thing. So that could be something like de-animalization. So you make it to an object rather to an animal. So that's the first scoping question. What kind of concepts and how broad should that be? And I think there is a need to be as broad as possible, at least at the start of the discussion. The second point in the ethical discussion is to see a tendency to only focus on what's new. So to say, well, for instance, in genomic selection, say, well, that actually is only a little bit accelerating what we have done for ages. And even with genome editing, well, yes, it's a bit new, but relative. Whereas the ethical discussion could be much more broader than only that sometimes tiny part that is strictly spoken new. So even if you have a technology that is in line with something going on, it still could imply that it will speed up a process or have an impact on what's going on, Or, and that's sometimes frustrated if it's your technology, it could be that this is just the tipping point where you get public discussion. So that you say, well, this is the small next step, which may be in itself not a huge change, but that is the reason why it's embedded in wider discussion on, for instance, livestock farming. And that also links to the final one. You could say what we're discussing here is only about cutting edge steps in breeding technologies. That's it. So we also have to limit ourselves to the discussion. At the same time, especially if you start to discuss with a wider audience, you immediately end up with discussions about where do we stand with livestock farming in the next 5 to 10 years. How does it fit to a global trade? How does it fit with small farming? So it's at least important to be aware that what we're discussing here today is embedded in much wider debates, like the things on global hunger or climate change. So if we try to do this, and also after the break to broaden up the discussion, unfortunately that doesn't make the discussion immediately easier, and it also not always lead to easy answers. At the same time, that's not, I would say, a problem of the ethical discussion. It simply is a reflection of the complexity where we have to deal with. And it also allows us to be, what I mentioned, as professionals, be responsible in the sense that we have responses to questions from different types of stakeholders. So also here, I think that there are a lot of stakeholders involved. But the point is, of course, that there is not a simple line of where to include the stakeholder. So it's important in terms of co-creation that you really try to develop technologies together with others, that you're aware of the broader set of interests and values. But also, and I think that's already a nice thing in both the BOVREG and the Geronimo project, to prevent that you have this type of discussion only at the end of pipeline, so that you already said, well, we have done wonderful research for 4 years, and then say, okay, well now we have to include society, because then, well, it's never too late, but quite often you have made a lot of discussions in your research which make the discussion with others much more difficult. The problem, or at least the importance of both stakeholder and public engagement is that, at least from literature, and I think there are strong reasons to say that if you be focused on responsible research and innovation, public engagement is not an add-on, but really an essential element. And the other point is, if you try or remain only in your own bubble, you end up with probably great research, but you easily have a very small focus. And I do not know whether you can recognize the picture. It's from the COVID period, and when we had the football matches without the audience, it's possible, but it's no fun. And I think that's a bit of where we need to go with research as well. It's possible to develop all kind of interesting steps within also animal breeding, but if you take stakeholders in a wide sense, so both professionals and general public into account, it really makes a difference, and you're playing a different game, and most of the time a better game. So then the question, of course, why? How to use them, and this is not a full list, but you could say you can use them as a source, so you can have an idea of how do people think about this, to enable them. So as I said, there is not so much public discussion about animal breeding at this moment going on, but you could say, well, actually, it is important that there is a broader discussion about what we're doing here. But then we need to enable people and give them the concepts, the words, to also help us to reflect and discuss. And another part is really to enable participating at a level of research, at a level of policy making. So this is how we try to do that within the Geronimo project, really to be aware that there is the difference between scientific knowledge, ethical challenges, and societal challenges, but that they are integrated. Here we also deliberately make a distinction between the societal and the ethical issues, because it could be that within society there are arguments highlighted which probably always have an ethical issue, but maybe from an ethical perspective, maybe not the most prominent, or the other way around. It could be that something is not mentioned in society, which from an ethical analysis is still something that you could highlight to be important. Still, it's not easy. Especially if you have general public, we have all kinds of social media which sometimes very simplify discussions, and we are doing great jobs, and then someone still says, well, I don't buy it. So that's a difficulty. At the same time, here again, maybe this nice cartoon shows also a little bit that these haven't talked before. Yeah? So if you try to take others into account from the very start, do not know whether you can prevent it, but it helps. The other thing, and that may be also a challenge for research and science, it's important to take public perspective into account. At the same time, it's important also to be aware of where you stand for yourself. So it's not the idea of public engagement is not that others fully develop or determine our agendas, but at the same time, it's no reason to exclude them from the start. So there really would be a kind of two-way engagement. It's not only us telling them what they should know, or us telling them what will happen in 5 to 10 years or 10 to 15 years, but really to enable discussion, to find a more way of dialogue. Two examples of how we did it in the Geronimo and the Bovrek project. One, in the Geronimo project, we developed and performed 8 focus groups in 4 countries, in France, Germany, the Netherlands, and Slovenia. The idea behind the 4 countries is partly because they're consortium partners in Geronimo, but also that the hypothesis that it's also important to find national or regional arguments. Science is very much on the global level. You see that policy is quite often on a European level, where a lot of public, but even also stakeholder discussions are embedded in specific context. In the Netherlands, we have problems with nitrogen emissions, which heavily influence all kinds of breeding. If you are in a country where organic farming is very much on top of mind, it influences that discussion. So that's what we just finished before summer, currently working on the results. And for instance, also as a spin-off, we did with the National Science Week last October, a dialogue with families on human-animal relationships and technology. Just that's a very open this way, more in terms of enabling them to have discussions. Another part in the Bovrek project, Donald and are here as well, we developed the Demox game. If you want to know more, exactly, the result is there, and Donald and Bruce will be here during lunch as well, so please consult them. But it's also a way of bringing in the knowledge from the Bovrek project into a public discussion. But again, that's not only for those out there, but it also proved to be very interesting to have this discussion among scientists and wider professionals. So what we'll do after the break is actually to have an idea of what kind of ethical and societal issues do you think to be important, but also, and that's the focus for this session after the break, how could we integrate them into policy? That could be company policy, but also on a governmental level, and what is necessary to improve that process. So to figure out what kind of hurdles you see and how could it be done. 2 rounds, so we have round 1, 35 minutes, you will have a scheme, I will show that later on. Define in your small group a number of topics where you think this is related to ethics in society for the GENE-SWitCH or the Geronimo, or maybe BovReg as well. The question, are these sufficiently addressed in policies? Yes or no? And why not? And then try to list what you think are hurdles in order to deal with these ethical and societal issues. Try to be as specific with the hurdles as possible, because the idea is that we switch, so you get the hurdles from the other, and you are asked to, in round 2, explore ideas of how we could overcome these hurdles and formulate best practices. And prepare, and that's why we have 5 minutes more in the second round, a short presentation where we can exchange our ideas at noon. The acknowledgements, because I'm standing here, I already mentioned Koen and Donald Bruce, but there are a lot who made this possible, so many thanks for them. I quickly go to this scheme, because then I do not need to introduce that after the break. This is what will be the scheme I'll ask you to fill in. These issues need not to be 4. If you have 5, that's fine as well. If you have 3, so that's still open. So think about your own, and try to fill in this, as I said. But especially these hurdles are do not say the most important, but at least would be nice as a final result after those 35 minutes. And then in the second session, you get the hurdles from the first group, and then the question is, how could we improve on that? Any questions so far? Then it's 10:30 and time for a break. So I look at the show. Yes? That's just here. Okay, so if 15 minutes break, and then given the group, so we'll split in 2 groups. So let's say 1 table over there, 1 over there. Feel free to sit wherever you like. And also online there will be breakout sessions that you will, yeah, great. 2 groups online as well, perfect. Okay.
there's this strike here, so some people will leave.
Oh, yeah,
that's why.
yeah, no, that's fine.
Okay.
Nicole and I will have a short presentation on, or not presentation, but giving back what we take from the two sessions. And
Yeah. 2 sessions.
Yeah, sorry, the 2 tables.
Oh,
so,
yes,
so
but we also have.
Yeah.
Oh, okay. Do you hear me? Hello. Do you hear me? Do I need microphone? Okay, so we weren't that many people in the online session. So it was Ellen, Tina, and we 3 ladies. But it was a nice discussion. Anyway, so we found 2 issues. One was the transparency on agri-food systems. And the second was about the education about genome editing and defining or missing information about it, about the new technologies. The relevant stakeholders are mostly everybody in the sector, but also a big focus on the younger generations and consumers and general public. But of course, we also have to network with the policymakers. So if we go from transparency, we thought that it is addressed a little bit about, with the labeling systems, but it needs further standardization. Maybe another system which is very easy to understand by the consumers, like this DOPs, the protect geographical indications, if needed. And at EU level, I think we should definitely involve the supply chain for these policies if there are going to be regulations to solve these problems. So we should come into a joint agreement within Europe, so not national or regional basis. But the main hurdle that prevents the inclusion of these in the policy is the lack of collaboration between different stakeholder groups. So also even leading to refusal of even discussing it. For the missing information or misinformation about the technologies, we think it is somehow addressed in the governmental policies, but it needs better distinguishment of different technologies. At the moment, it's under GMOs, and it needs, I think, better distinguishment. And it could be further embedded, but more on a holistic point of view. Maybe, I don't know if somebody wants to explain it better. But there's quite a lot of lack of knowledge exchange, and the technologies, there's no technology to identify gene editing at the moment on the product level. So how are these addressed? How could these be addressed? Well, mostly with a lack of collaboration. We thought there could be maybe some cost actions, some other projects that is mostly focusing on the networking or, joining different stakeholder groups in a wider, broader coverage. And science-based communication, which is quite simple to explain, simple to understand by the young generations especially, but in general the public. And for the gene editing, of course the technology needs to be there to identify gene editing, so that it could also make it easier maybe for the consumers to understand if we have it here or we don't have it, since still the imported foods are also coming from an origin of gene-edited plants and animals or not. So this is an important technology to invest in. And we also have to address the hurdles with scientific evidence, especially focusing on the future challenges, like how we are going to feed the world with organic production and small, very small, or very, like romantic, idealized farming systems. So that's why we have to cooperate with interdisciplinary researchers and areas like human geneticists, as well as communication a lot. And yeah, I think, did I miss something? Yes. I don't know if we did it right, but
Yeah, so the first group I had on the table discussed quite a range of topics, but we ended up spending most time discussing increasing efficiency as a breeding goal and a breeding goal with a societal relevance, namely not just to increase profits in animal agriculture, but also to help feed the world and at the same time limit the environmental impact of animal agriculture. This topic led to the identification of quite a range of stakeholders as well, including companies, where a question was, so is every company equally able to make use of this increase of efficiency, or are only the large companies able to do that? Consumers may profit from having access to good and cheap foods. Animals are were also mentioned as relevant stakeholders. And well, it was considered an important issue, an important trait to work on for these societal reasons, but we struggled a little bit to identify the ways in which improving efficiency was already also on the policy or legal agenda. Well, it was clear that there is research funding for projects aiming to improve efficiency by means of other breeding methods than genome editing, at least. There are already, through the Common Agricultural Policy, some stimuli to breed feeds locally that animals can consume. But well, there may be more ways to include this in policy and law. Well, one of the options that we discussed was stimulating longevity as a breeding goal. So animals, at least who produce other products than meat, are more efficient in that way, and trying to stimulate circular production, so the use of waste streams as feeds. But there are also quite some hurdles to achieve this, including consumers who are concerned about the environmental impact of animal agriculture may prefer other solutions than improving efficiency by breeding, in particular just switching to a diet which contains fewer animal products, or even to an all plant-based diet. And so, whereas consumers who just want to keep on eating meat may not be concerned about these societal issues. And then in the second group, we continued this discussion. So what can we do to prevent to conquer these hurdles? And it was mentioned that well, maybe the demands on breeding are not always reasonable, in the sense that, well, efficiency is already quite high, and we all have all kinds of other breeding goals as well, which need to be weighed against efficiency. But the super animal, which addresses all these breeding goals to an optimal extent, doesn't exist. And maybe in the developed world, efficiency is not the breeding goal that should receive the most weight. There are also all kinds of other breeding goals, including welfare traits that may deserve priority. But in developing countries, efficiency may be an important goal to focus on. Although it's currently quite challenging to improve the efficiency of animal agriculture in developing countries, because the infrastructure is missing to implement breeding techniques, but also the knowledge to collect data in a suitable way, and all kinds of hurdles in that respect. But there were also practical steps and stakeholders that could be could help to address these issues. So the main message was we need to sort of collaborate more with local people in developing countries, and not just the universities and other knowledge institutes, but also farmers, people on the ground, to see how what their needs are and how efficiency could be improved in ways that suit them. So that was the main message, I think.
Thank you so much. Well, the table I hosted in the first round, we actually had two big issues. One was all kind of issues that are linked to the high-tech solutions we use in breeding, and then specifically also on genome editing. And the other was more specifically on the use of animals, also in terms of animal experimentation. Where the first was, of course, the existing legislation we have with regard to genome editing, but also in terms of, well, you could say there are all kinds of frames and ideas and misinformation on what we think is going on in breeding, and then the we is not so much the we as we are here, but the outside world, and where maybe the main hurdle was we got the impression that legislation, or at least policy, is sometimes designed based on information that is maybe 20 years old, and how could we improve that? And the other point was how the hurdle with animals, and especially experimental animals, how could we come to a situation where that is more tailor-made to the demands that are specific to using animals as a goal animal. Yeah, so the EU directive is very much designed as a way to regulate lab animals for mainly medical or safety issues, rather than for the work which is going on in breeding. That last point didn't make it in the second round, because already the first one took a lot of interesting discussion. In a very practical way, the idea was how to overcome that problem of building or designing a policy on, let's say, old information of disinformation. First was to say, well, we need to do education starting already on a young age. At the same time, in the discussion, then it was also a little bit not so much nuanced that was not important, but to say there should be more, because what do you need to educate and who? And then was okay, of course, science needs to play an important role. But media plays a role as well, sometimes in a specific way of framing, but it was also mentioned as one of the stakeholders in practical steps. For instance, examples with television programs where farming and farmers are portrayed in a not necessarily only positive way, but that you start to attach to what is going on. And I think that was one of the main messages, was let's try to communicate in an honest way. So there was a little bit of, where is it, the discussion, for instance, on the small picture here on the GENE-SWitCH, where you have the farm with the pigs outside, low density, which looks nice, but which is not the real picture of what's going on and what we're doing. So also to maybe a point for us here, but also with all kind of stakeholders, if we start to communicate, try to tell the honest picture. And that's one. And the other point was to say that might not convince everyone. And that's maybe another concrete step that we say we try to be explicit as possible about where you stand for. So if, and I think it was mentioned as well, if we have a discussion with others who really are in favor of a animal-free diet, that's a legitimate standpoint, but there you cannot convince someone telling that in terms of animal welfare what we're doing is quite good. And so try to be both as transparent in the way of how we communicate with each other, but also don't come up with too nostalgic pictures that only would add to that hurdle we defined. So that was it. And the interesting thing is, of course, that we collected a lot of interesting discussion that we didn't further explore. Although we try to be as concrete or practical as possible, it's of course only scratching the surface. But what we hoped with this was to make at least the societal and ethical issues a little bit more practical, and then immediately you see that it's not a kind of single owner. You cannot say, well, now you are a farmer and now you have to deal with the ethical issues, or that I can look to you and say, okay, well, you're a science, now you have. So it's really a joint activity, which on the one hand makes it easy to step aside and say, well, it's not me, it's you. So collective action problems also are a problem, I would say, in dealing with the ethical issues, but that we really need each other and also reach out to other stakeholders to deal with its question. We had a formal closing of this session. I do not know. Do you need, or will you?
Stay here.
Yeah.
You can stay here. I think. So it's like the witness, but we should give to. Right? Something like that. We didn't prepare anything. Did you prepare any statement?
No, just thanks all the people today that contributed to the workshop and to the exchange we had, and finally to get these conclusions. It was really nice to mix the two projects, I think, and to get different point of view, to see also how Geronimo is following what we have done for GENE-SWitCH in a different way, but complementary way. And it's nice to see that what we have done is still living with others, and also with us. We are still working on GENE-SWitCH after, I think, the end of the project, but it's also followed by others with other questions, and this is really a good aspect of the clustering of these projects.
Same here. I think we made a very good job, and we were able to keep in the big picture and to co-build with others the big picture. This is continuing, and Geronimo is taking the lead now, let's say, for going further, also with other projects that will follow. And also from my side, of course, again, a big thanks to everybody who contributed and was here and presented, and also in the whole course of the GENE-SWitCH project. Thank you.
Yeah, just 2 words before lunch. No, I think we really see how throughout time, and thanks also to Eurofund, the different projects that might be interested in different species, and up to, I don't know, a few years ago, it was hard to imagine a sort of talking between species. Now it becomes possible. I think there are really bridges between projects, and a lot of exchange, and a lot of exchange of methods, but also of ideas that I think it's something extremely constructive for the future. Thank you.
you.